Hello everyone and welcome. In this video we're checking out variant 1 of October-November 2024 paper 2. Before starting, make sure to check out the free notes I have available for you in the comments section down below. I've uploaded new notes for chapter 10, infectious diseases. Now let's go ahead and get started. Question number 1. Animal cells, plant cells, and prokaryotic cells have similarities and differences in their structure. Table 1.1 lists 5 organelles found in cells. Complete table 1.1 by placing a tick to show whether the organelle is present in animal cells, plant cells, and prokaryotic cells, or a cross if the organelle is absent. Okay, put a tick or a cross in every box. In the first row has been completed for you. A nucleus is present in animal cells, plant cells, but definitely not in prokaryotic cells. A large permanent vacuole is only present in plant cells. Animal cells have temporary small vacuoles. Prokaryotic cells don't have vacuoles. Rough endoplasmic reticulum. The rough endoplasmic reticulum is found in both animal cells and plant cells. Prokaryotic cells do not have any membranous organelles, therefore X is the answer here. The Golgi body is found in animal cells and plant cells, but not in prokaryotic cells. Centrioles are found only in animal cells, not in plant cells, and not in prokaryotic cells, and so are centrosomes. Now let's go ahead and move on to letter B. Now, figure 1.1 shows the section through an epithelial cell found in the digestive system of an animal, and the cell is specialized for absorption of digested food. So this will probably be part of the small intestine in the ileum, where the cells are obviously going to have microvilli in them. And so P over here is the microvilli, and Q is the mitochondrium. So you could see the structures labeled P and Q in figure 1.1 are involved in the absorption and digestion of food. Could you name the structure labeled P? So that's microvilli. Okay, explain how the organelle labeled Q is involved in this process. Now, the organelle labeled Q over here is mitochondria, and mitochondria carry out had aerobic respiration, and so they release energy. Energy is required for, obviously, the absorption of food by active transport, where food is going to move from a low concentration to a high concentration against their concentration gradient via protein carriers that need the energy to carry out a conformational change in shape. And let's not forget things like endocytosis, basically cytosis. Okay, these all require energy, so let's go ahead and write that down. Okay, so mitochondria, which is the site of aerobic respiration, which is obviously important to produce ATP. And obviously this ATP could be used. You could say here, for example, it's hydrolyzed, releasing small packets of energy. For absorption of, here he says, food, digested food. Obviously by active transport. And you can define active transport by the definition that I gave you just a minute ago, which is movement from lower concentration to higher concentration against the concentration gradient, where the energy is required for the carrier proteins to carry out conformational change in shape. And then you could also say cytosis, which, to be more specific, obviously endocytosis. Now let's move on to question 2. In mammalian circulatory system, red blood cells travel through different types of blood vessels as they pass from the heart to the respiring tissue and back to the heart. Now, this shows the types of blood vessels which red blood cells travel in the circulatory system. And he's saying here, okay, so let's see the question. Complete figure 2.1 by writing the names of the missing types of blood vessels through which red blood cells pass. Okay, so we need to just complete the circulation here. So you can see heart goes to arteries, arteries to something, to capillaries. Well, what is the link between arteries and capillaries? It's the arterioles, which are the smaller arteries. And then capillaries to veins, we need venules in the middle. Okay, moving on to letter B. Water is the main component of blood. It has an important role in the transport of substances around the body, and figure 2.2 shows the ionic compound sodium chloride dissolving in water. So you can see we have how the dissolving happens is that the ion is going to be encapsulated by water, surrounded by the parts that are going to be able to basically have the same charge as it. For example, we know that water is a polar molecule and that it is going to have a partial positive. The hydrogen ions are going to have a partial positive charge, the oxygen will have partial negative. So you'll find that the hydrogen ions, or hydrogen bit of the water, is going to be closer to the chlorine, and that they're going to form hydrogen bonds. So here he says, explain how water acts as a solvent for sodium chloride. So to talk about how this happens, we just describe everything I just said just in a more basically in a more proper manner. So let's start with how water is polar and it's a dipole. So it has a partial positive bit and a partial negative part. Okay, so hydrogen atoms and partially negative oxygen atoms, and then from here you can say something like the hydrogen atoms, which are partially positive, are going to be attracted. So we can talk here about the encapsulation. So water molecules obviously, when they encapsulate the molecules, the ionic bonds between the sodium and the chlorine are going to break, okay? Breaking the ionic bonds. And then you could talk about how, now the negative part is attracted to the positive part and so on. So negative or delta negative oxygen is attracted to the sodium ions, and so it circles it by basically, whereas the partially positive hydrogen atoms are attracted to the negative ions. Okay, so this is it for this question. This is how water acts as a solvent, because remember, it's a polar molecule, and obviously like dissolves like, and how it acts is basically it forms this capsule or the hydration shell, and then there will be the breaking of the ionic bonds and the formation of these kinds of bonds here between the hydrogen and the chlorine and the oxygen and the sodium. Moving on now to C. Figure 2.3 shows a Galapagos penguin swimming in water. So we have a penguin swimming in water, and penguins are birds that live on land but spend a lot of time swimming underwater hunting for food. Penguins can remain underwater for up to 20 minutes. During this time, they do not breathe, but their tissues continue to respire. And he says here that hemoglobin in red blood cells of penguins has a higher affinity for oxygen than hemoglobin in other birds that do not swim underwater, which makes sense because there is not as much oxygen underwater. Figure 2.4 shows the oxygen association curve for a bird that does not swim underwater. Draw a line on figure 2.4 to suggest the position of the oxygen association curve for the hemoglobin penguin. Okay, the penguin hemoglobin. So to draw the curve, what we need to do is we just need to draw the same graph, but over here to the left. Okay, so let's basically do that. Same shape, same everything, but obviously it's just a little bit higher. It doesn't need to end at the same place. I don't think it needs to end at the same place. It just needs to be that it's basically higher up. Okay, so let's just raise this part over here and just make it higher. Obviously make it a smooth curve. Don't do any of these lines because you're going to be detected on having any breaks in your lines. So just make it a smooth sigmoid graph that is a little bit to the left because of the higher affinity. Now, penguin hemoglobin is sensitive to decreasing in pH caused by an increase in the carbon dioxide concentration in the blood. Explain how the decrease in pH penguin hemoglobin, and suggest how decrease in pH affects penguin hemoglobin, and suggest how this helps the penguin to swim underwater for a long time. So he said here that the penguin hemoglobin is sensitive to a decrease in pH, which means if carbon dioxide concentration increases, the hemoglobin is going to be sensitive to that decrease in pH, and suggest how this helps the penguin swim under for a longer period of time. Well, remember, oxygen here has a really high affinity to hemoglobin in the penguin, and so since there's a really high affinity, there's a chance that obviously oxygen just stays attached no matter the pH. Now, since the blood is affected by decreasing pH and the penguin is sensitive to it, this means that oxygen can be released, and carbon dioxide can bind to the hemoglobin, which is very important because remember, it's not just about carrying oxygen to the respiring parts, it's also about expelling the waste material like carbon dioxide out of the body through the lungs. And that's why we need to we need this sensitivity to pH. Okay, so we need the sensitivity to pH because the decrease in pH, this is going to be decreasing hemoglobin affinity to oxygen. So oxygen can be released, which allows, now it's not really CO2 that binds directly onto the hemoglobin, it's hydrogen ions that bind onto the hemoglobin, which is basically the Bohr shift. So I would advise you guys to describe the Bohr shift just to make sure that you guaranteed the grade. So which allows hydrogen ions to bind to hemoglobin so that CO2 can be expelled from the body, and obviously just basically be able to continue respiration normally, so to continue now. Obviously you can here link the decrease in pH with the hydrogen ions because it's very not subtle for you to just go, oh, decrease in pH allows hydrogen ions. Okay, a decrease in pH has to do with hydrogen ions. So we can say basically that decreasing pH means higher CO2 concentration, which is why the hemoglobin has now a higher affinity to hydrogen ions. Higher hydrogen ion and please remember, if you have time in the test, describe the Bohr shift, it will give you more grades. So hemoglobin has a higher affinity to them. Okay, moving on to D, the heart rate of a penguin decreases while it's swimming underwater. The heart rate is regulated by a group of specialized cells in the wall of the right atrium, which are obviously the cells of the synovial node. Now, the activity of these cells is modified by nerve impulses. Name the group of specialized cells in the walls of the right atrium that regulate the heart rate. They are the cells that basically form the rhythmic contraction, which are the cells of the sinoatrial node. Now, moving on to 3. Figure 3.1 is a photomicrograph of a transverse section through the wall of the bronchus in a gas exchange system, and basically identify the tissues J and K and suggest how the wall of the bronchiole differs from the wall of the bronchus for these two tissues. Well, sure, okay, first he needs you to identify what are J and K. So you could see how J is a thick part, and remember when it's thick and it has these cells, the chondrocytes, this is definitely cartilage, whereas K, it looks, like a smooth muscle. Okay, you can see how it's basically right next to it, and it's a very long part. I think, if you've dealt with a microscope, you'll be able to identify them. But however, if you feel like you're not comfortable with identification, please go back to the book and review the images there. So now K is going to be the smooth muscle and J is cartilage. And what's the difference? So this over here is the bronchus. Okay, and he's looking for a difference between bronchiole and bronchus. Well, bronchus has cartilage. Bronchioles do not have cartilage. Bronchus have these blocks of cartilage, whereas bronchioles do not have cartilage. Another thing, maybe, let's think about. Okay, he's looking for the difference, and I think that identify and suggest how the wall of the bronchus differs from the wall of the. Okay, I think that we can just list one point here, and we should be good. So cartilage only in bronchus, definitely not in bronchioles. Okay, because I think it's pretty much the only clear difference that we have identified before. B. Tuberculosis, TB, is an infectious disease that affects the human gas exchange system, and the pathogen that affects the pathogen that causes TB secretes a protein that can be detected in saliva. Early diagnosis of TB is important in reducing the transmission of the pathogen, and scientists have developed a test strip for TB that uses monoclonal antibodies, and monoclonal antibodies are specific in their action. The test strip contains the following: mobile monoclonal antibodies that bind to one part of the protein secreted by the pathogen. So we have mobile monoclonal antibodies that bind to one part of the protein secreted by the pathogen. And he says here also that immobilized monoclonal antibodies, so they're just in one place. So figure 3.2 shows the simplified diagram of the test strip. This is the area where the test strip can be held, and then this is the control area, the direction of flow of saliva through the test strip. So this is going to be where I think the patient places the test strip in his mouth, a sample pad where the saliva is added to the test strip. Okay, and then area containing mobile monoclonal antibodies attached to tiny gold particles, and then a test area containing immobilized monoclonal antibodies, and then a place where there is no antibodies, and this is where we're going to hold the test strip. Now a sample of saliva is collected and put onto the sample pad on the test strip, and he says here that the saliva moves up through through area 2, and the mobile and monoclonal antibodies are attached to gold particles. If these antibodies collect in test area 3, a gold line becomes visible on the test strip. So if the antibodies collect in this area, for example, this is going to be where a gold line is going to form, and then a gold line that becomes visible in area 4 confirms the test strip is working and that the results are valid. So if what's it called, gold line that becomes visible in area 4 confirms that the test strip is working and that the results are valid. I think the control area, immobilized, where are the immobilized? This is where the immobilized pathogens are. Okay, so let's continue. So the saliva is going to move all the way up here, and if the gold line becomes visible here, this means that the test is working. Now, state the name of the pathogen that causes TB. Well, TB is caused by Mycobacterium tuberculosis. Actually, it's caused by two pathogens, Mycobacterium tuberculosis and Mycobacterium bovis. I think we are, since they just wanted one, we can list any one of them. However, let's just make sure to write both. So Mycobacterium tuberculosis and Mycobacterium bovis. Okay, remember to write the genus capitalized, the species small, and the name in cursive, or if not in cursive, underline it. Name the part of the monoclonal antibody that binds to the protein form of the pathogen. Okay, the part of the antibody that binds is going to be the antigen binding site, the variable site of the antibody. So let's write this down, the antigen binding site. Okay, saliva is added to the test strip to test the presence of protein secreted by TB in the pathogen, and figure 3.3 is the result showing some of the monoclonal antibodies in area 3 of the test strip when a positive result for TB is obtained. So he's saying here monoclonal antibody attached to a tiny gold particle, and the protein secreted by TB attached to two different monoclonal antibodies, and these are the immobilized antibodies in area 3 to test a line. So basically this is area 3, and that these are the monoclonal antibodies. They're going to attach, but then they're going to get attached to also to the immobilized antibodies. So this is why the gold line forms. So use information in figure 3.3 to suggest why this test is specific for TB. Now, the test is specific for TB because obviously the monoclonal antibodies that we're using here are going to have an antigen binding site that's complementary to the antigen of the TB protein, okay, and it's a protein that's secreted only by TB. So it has a binding site for the which is complementary to the protein, and also we can mention that obviously the monoclonal antibodies here are very specific, and so they can't really bind to just any protein. They have to bind to this specific protein. And and lastly, why this test is specific for TB, I think that's it. We can just write this down since it's a 2-mark question. I think this will do. So let's go ahead and write that down. Firstly, the monoclonal antibodies have a antigen binding site complementary to the protein released by TB, and the binding site is very specific. So the antibody only binds to this protein by this bacterium. Now, this is a suggest question, so feel free to write down anything that comes to your mind. I think these are the most clear options. And then if you want to describe how they're specific and where they're specific, maybe describe the globular protein structure, this also works. Now let's move on to area 4. Contains different immobilized antibodies to those in area 3. Okay, and the mobile monoclonal antibodies bound to tiny gold particles will bind to those immobilized antibodies in area 4. If the test has functioned correctly, a gold line will be visible in area 4, suggests how the structure of immobilized monoclonal antibodies in area 3 differs to the structure of monoclonal antibodies in area 4. So what he's saying here is that the monoclonal antibodies are going to bind to immobilized monoclonal antibodies in area 4. So themselves, the mobile antibodies are bound to tiny gold particles will bind to those. So I will have to say that maybe the difference here, maybe we could suggest that, for example, while this protein over here binds to, so the antigen itself is going to be the one, the protein itself is the one to bind to those antibodies, and the antibody is the thing that's being bound to the other antibodies. And obviously he's looking for the difference in structure. So the difference in structure between different proteins, we can just list the differences in structure, so whether it's primary, secondary, tertiary, or quaternary structure. I don't think quaternary structure will be different. It's always going to be consistent of four polypeptide chains. Now let's go ahead and just write down that it's going to have a different antigen binding site. And let's suggest something, so different antigen binding site could be, for example, complementary to antibody itself, for example, antibody instead of protein, and then you can mention why they're different. So the difference in antigen binding site could be caused by difference in primary, secondary, or tertiary structures. And so, and obviously we can list how, for example, tertiary structures are different, or how secondary structures are different. We can mention things like the different bonds that are part of the tertiary structure, or the different shapes that can form the secondary structure, the alpha helix or the beta pleated sheet. But since it's again a 2-mark question, I think I'm going to leave it here. But please, in the exam, go back and add more information to your answers. Now, letter C. Vaccination is another way to reduce the transmission of infectious diseases such as TB. The BCG vaccine is used to help control the spread of TB. And this vaccine contains a weakened strain of the pathogen that causes TB, and the BCG vaccine stimulates the development of the antigen-specific memory T lymphocytes. Interesting. Explain how the memory T lymphocytes provide protection from TB in a person who has been given a BCG vaccine. Well, obviously, a memory cell is going to provide long-term immunity in that, and if the pathogen comes back again, it's going to stimulate a primary immune response. That's not really going to be a primary immune response, since it's not the first time that we face the antigen, and it's going to be a secondary immune response, and it's going to obviously be faster, it's going to be easier. The patient is not going to feel as tired because his body isn't working twice as hard to identify the pathogen in the first place. We already know the pathogen, and obviously since memory T cells are present, then they're going these memory T cells are going to identify the antigen pretty easily, and they're going to secrete they're going to stimulate the secretion of cytokines by the T helpers lymphocytes, because that's what T lymphocytes do. And then you could just mention anything regarding the secondary immune response, whether it's cellular immunity or humoral immunity. And let's go ahead and write this down. So as we mentioned, long-term immunity, where basically the long-term immunity stimulates a secondary immune response when the pathogen obviously binds, or pathogen's antigens are recognized by the memory T lymphocytes. Okay. And then you could mention how the T helper cells will secrete cytokines, and we can also mention anything in regards to, how it happens after cytokines are released. So for example, this could activate macrophages, or it could activate B lymphocytes, and so on. And so T helper lymphocytes which is obviously TB. The bladder is the organ of the body used to store urine. When cells divide uncontrollably in the bladder, a tumor develops, and this can lead to bladder cancer. And the BCG vaccine is used to help treat bladder cancer. Interesting. The BCG vaccine is introduced to the bladder, and the tumor cells take up the weakened pathogens in the vaccine, and they act as antigen-presenting cells. Okay, yes, so what happens here is that the tumor cells themselves, they take up the pathogen, and they act as antigen-presenting cells, and the name of the process by which the tumor cells take up the weakened antigens, well, it's going to be by endocytosis, or, phagocytosis, but I would prefer you guys just write endocytosis, because this is not a phagocyte, it's a tumor cell. Suggests how antigen presentation by tumor cells stimulates an immune response that leads to the destruction of tumor cells. Okay, so just basically talking about the stages of the immune response. So firstly, we have antigen presentation. This is going to be followed by clonal selection, then clonal expansion, no matter the types of cells. So since here we're dealing with T helper cells, I think we've established that, right? They didn't. But let's just basically talk about how the immune response is going to happen. So what's it called? Firstly, there's going to be antigen presentation. Now, since there's antigen presentation, some of the T lymphocytes are going to have receptors that are, complementary in shape to the antigens on the surface of the pathogen or the antigen presenting cell. And once that happens, this is the process of clonal selection. Now, once clonal selection happens, this is going to stimulate the cells to divide. So the cells that have now the complementary receptors, they're going to divide by mitosis in a process known as clonal expansion. Once these cells are going to divide, they're going to start producing cytokines if they're T helper cells. And if they're T helper cells, they produce cytokines. These cytokines stimulate the different cells of the immune system. If they're T killer cells, they're going to produce toxins like hydrogen peroxide, and these are going to be released and released to the antigen presenting cell, causing them to be killed. If they're again T helper lymphocytes, they produce cytokines. Cytokines could stimulate B lymphocytes to produce plasma cells, which produce antibodies. They can also stimulate B lymphocytes to become B memory cells. They can also stimulate macrophages to attack the pathogens more vigorously. They can stimulate, what else could they? I think that's it. Okay, they can also stimulate T killer cells. So let's go ahead and write all this down. So some T lymphocytes have receptors complementary to the antigens on the antigen presenting cell. This is known as clonal selection, which is when a cell has a complementary receptor. Now, after clonal selection happens, this stimulates lymphocytes, or T lymphocytes more specifically, by mitosis, which is obviously clonal expansion. followed by how the T helper cells release cytokines, and how the T killer cells produce toxins that kill the antigen presenting cell or the tumor cell. And the cytokines act as cell signaling molecules and stimulate. You can mention any cell here, like B lymphocytes. I don't know why I wrote beta, it's B. Okay, B lymphocytes to produce plasma cells, so for example to divide and produce plasma cells. And antibodies by plasma cells. And then you can also write how, for example, they can stimulate other cells like macrophages, and T killer cells produce toxins. You can write down the examples of toxins. And I think that's it. Let's move on now to 4. Figure 4.1 shows the structure of sucrose, a disaccharide produced by plant cells. Name the covalent bond that joins the two monomers. Well, it's a glycosidic bond, and it's specifically in the case of sucrose, it's supposed to be it's a 1,4 alpha glycosidic bond, but obviously this depends on the textbook you're using. In the Cambridge book, we talk about a 1,4 alpha glycosidic bond. Let's just call it a glycosidic bond. 2. Sucrose is hydrolyzed by the enzyme sucrase in the human digestive system. The products of this hydrolysis reaction are the monomers of alpha glucose and fructose. Complete the diagram to show the hydrolysis of sucrose to form alpha glucose and fructose. Okay, so the hydrolysis, remember with hydrolysis we need H2O. After H2O we can just go ahead and split these two. So here we're going to break these and basically place an OH instead. So firstly we have alpha glucose. In alpha glucose we have down, down, up, down, and don't forget about the side chain CH2OH, and obviously don't forget about fructose. Now he said here to complete the diagram to show the hydrolysis of this stuff. Okay, so we can just go ahead and continue here. And then actually no, here we have an H, no H. It said structure of fructose, and then OH and H. Or do we have an OH? Actually, we do have an OH. It's just that it goes away right here. Okay, so let's continue. Now that we've done the structures, we can label them. So this is alpha glucose. This is fructose. Now let's go ahead and continue. Okay. B. Plants transport sucrose from a source to a sink, and he says here scanning electron microscope SEM of a transverse section through a plant tissue is used to transport sucrose. Yes, okay, so it's used to show the structures. Yes. Okay, so name the structure labeled X. So since you could see the structures labeled that are going to transport sucrose, we know they are part of the phloem. They are the sieve tube element and the companion cell. Now the sieve tube element is going to have these end plates. So this is the sieve tube element, and to be more specific, we labeled X, so it's the end plate. Or you could just say sieve plate if you like. Now 2. A scientist carried out an experiment to study carbohydrate transport in the stem of a woody plant. Figure 4.3 shows a plan diagram of the transverse section of a stem studied by the scientist. The position of the xylem tissue in the stem is shown, so we have a pith, outer stem tissue, and xylem tissue. And it says the scientist carried out a set of experiments using plants of the same species. And he said here, in each experiment, a ring of tissue was removed from the outer stem of the plant, but the xylem tissue was left intact. and this is shown in figure 4.3. So he removed a part of the stem, but he kept the xylem tissue, so the region of the stem where tissue was removed. Now, this mass of the carbohydrate transported from the lower part to in the stem in 24 hours was recorded, and obviously because he removed the phloem, this means that there's no carbohydrate by the end, I think, of the 24 hours. And in each experiment, a different percentage of the outer stem tissue was removed, and all the variables were remained constant. And this is shown in figure 4.1. So the percentage of the stem tissue removed, the higher the percentage, if it's like 100%, there will be zero mass of carbohydrate transported to the lower stem in 24 hours because there is no translocation. There is no phloem to carry the carbohydrates. Now explain the results shown. Okay, so firstly, let's just explain it like babies, because there's 4 marks here. So let's just describe it in mundane detail. Starting out with, these are carbohydrates. Carbohydrates are transported by the phloem in translocation. There is no translocation when there is no phloem tissue. You can mention, for example, how when you reduce the amount of phloem tissue, this reduces the amount of translocation, and how as the percentage of the phloem tissue removed increases, this reduces again the translocation, or this decreases the amount of carbohydrate being removed. And then you could also give examples like this one, which is very important. When there's no phloem, there is no translocation at all. And you can also say that things like, what else could we mention? I feel like this could get you the 4 marks, but you can just continue by mentioning other things like how this was left for 24 hours, and each stem the mass of the carbohydrate transported to the lower part. We can mention, for example, the xylem tissue is the only thing left. The xylem does not transport the carbohydrates. We can also mention, for example, I think that's it. Okay, so let's go ahead and write this down. Okay, so carbohydrates are transported by translocation in the phloem. You can mention some points like by mass flow. And then you can mention here how as more of stem tissue is removed, this removes more of the phloem. Since obviously here outer stem tissue, since the phloem is in the outer stem tissue, since the phloem is in the outer stem tissue. Now that we're done explaining in general, we can refer to the results. So as more outer stem tissue is removed, there's less carbs transported or moved by translocation. And how, for example, how carbs only move in phloem, not in xylem. And you can mention the results here. For example, when it moves to 13 to 67, the mass of carbohydrate transported in the lower part of the stem in 24 hours in milligrams decreases from 774 to 597. So I will leave this up to you. You can write any example, but it's very important that we list this guy. So at 100%, when all phloem or outer part of stem is removed, so we're talking 100%, there is 0 milligrams of carbs were removed because the entire phloem was removed. Okay, were removed. Now C, sucrose is a sweet tasting sugar found in many foods. Some people become ill when they have sucrose in their diet. Okay, so it's a sweet tasting sugar, and some people become sick when they have sucrose in their diet, and these people have a gene mutation in the gene coding for sucrase, cannot hydrolyze sucrose in the digestive system. So since they have a mutation, this means that their sucrase is probably non-functional and does not work, and so it cannot hydrolyze sucrose. Scientists studying the DNA of people with this condition identified a deletion mutation in the gene code. Remember, deletion mutations are very dangerous, and so are insertions, because If you just insert one nucleotide, you can result in a frame shift, which basically results in a completely non-functional protein. Scientists studying the DNA to suggest and explain why the person with this deletion mutation cannot digest sucrose. Well, firstly, we can say how their sucrase is non-functional. And then we could say why, because it does not have the right structure. Since structure is changed, you can mention how the structure is changed, maybe primary structure changed. And how, since the primary structure is changed, this also changes. Tertiary structure, since maybe, for example, you don't have the right amino acids, so some bonds can't form. And you can write, obviously, the protein does not form the right structure. Then this obviously results in an active site that's not specific or non-complementary. So the active site is no longer complementary to the substrate sucrose. Then we can mention how deletion mutations work. So a deletion mutation means loss of one nucleotide, two nucleotides, it could be three, it could be more, so loss of one or more nucleotides, which could lead to missense mutation or nonsense mutation. Remember, missense means that there's simply a change in the amino acids. Nonsense means the result is a stop codon shows up. If a stop codon shows up, this means the protein is cut short and it's 100% non-functional. Now that this mutation has happened, it's definitely changing the sequence of nucleotides. And obviously you can explain the missense and nonsense. Missense mutations result in a changed sequence of nucleotides. Again, I erased this so that the paragraph is more cohesive. Missense mutation results in a change in sequence of nucleotides. Whereas nonsense mutation brings a early stop codon, which obviously is not good. And remember, these mutations as well as the deletion could result also in a frame shift. Now, obviously this changes the codons, changes the amino acid being coded for, and I think you can, imagine how you could continue the question, writing down. You have a lot of space to write down actually, so use this space wisely in a test. Now, I think this is more than enough information. Let's move on to question 5. Trypsin is an enzyme which catalyzes the hydrolysis of casein, a protein found in milk. Milk that contains casein has a cloudy white appearance. As the casein is hydrolyzed by trypsin, the milk changes in appearance to clear, transparent, colorless solution, and the student carried out an experiment to investigate the effect of enzyme concentration on the rate at which trypsin hydrolyzes casein, and the student added a solution of trypsin to a sample of milk and recorded the time taken for the milk to become transparent. The student then repeated the experiment with different concentrations of trypsin. All other variables were kept constant. Figure 5.1 shows the results of the experiment. 5.1 shows the results of the experiment. So you could see time taken for milk to become transparent and percentage change in concentration. So here In the question, when the concentration of trypsin changes from 2 to 4%, the time taken for the milk to decreases by 48%, whereas you need to calculate the percentage decrease in the time taken for the milk to become transparent when the concentration of trypsin increases from 0.25 to 0.5. Write down your answer to the nearest whole number. Well, it's pretty simple. You go here to the concentrations, you go to 0.5 right here, and then you go to 0.25, which is going to be at the center. So 0.10, 20, 30, 40, and 50, whereas 0.20 is here, 0.25 is in the middle. So now going to 0.25, you're looking for the time. So go all the way up, and look for the time, which you will find to be... Okay, did they even record this point? Yes, it is exactly this, it's x. Okay, so you have 360 to 400, and we're basically counting, I think, we're skipping by 40, which means each of these blocks is 4 units long. So if this is skipping by 40, this is skipped by 20, 380, 384, 388. Then, over here at 0.5%, we have the point over there, which is going to be 244, 48, 252, 256 right here. Okay, so we have change in time from 256 to 380, so it's going to be percentage decrease is going to be old minus new over old. So 380 minus 256 divided by 380 times 100, which is going to be equal to times 100 is equal to 32.6, which is approximately 33%. This is to the nearest whole number. However, no, it's actually 388. Oh, okay, 388 instead. So 388 minus 256 divided by 388 times 100, and that's going to be 34.02. So to the nearest whole number, it is 34%. Explain the results shown in figure 5.1. So to explain the results in figure 5.1 is basically to explain this exponential graph. So as you know, concentration of trypsin is going to be the limiting factor for the rate of reaction. As you increase the concentration, it increases the rate of reaction and hence reduces the time taken for calcium to be destroyed. So let's write this down. Concentration of the enzyme trypsin factor of rate of reaction increasing it increases the rate, hence decreasing the time to hydrolyze. Okay, so and obviously to explain the results, time taken for milk to become transparent, percentage concentration of trypsin. You could see how we could list some results. Okay, like for example here that's given to us, and here, okay, as for example, %. And we can go to the results here. For example, at 2 it's at 100 and I think 116, whereas at 4 it's only 60. I think in seconds. Yes. You can also mention the type of graph. So the graph an exponentially decreasing relationship, which basically you can simply say it's a disproportional or an oppositely proportional since we're explaining here increasing the concentration of enzyme leads to more collisions. You can first say more active sites and hence more collisions. Between the enzyme active site and substrate binding site, and so more successful collisions. Greater formation of substrate enzyme substrate complex. And since there's a greater formation of enzyme substrate complex, then obviously this results in a greater rate of reaction, as well as maybe we could say that the graph never plateaus. So enzyme concentration remains a limiting factor for rate of reaction, all the way to 4% or more. We don't know what is the limiting factor exactly. Now moving on to B, trypsin has the potential to be used in a wide range of industrial processes. Some scientists investigated the effect of temperature on the activity of trypsin immobilized on the surface of materials and trypsin free in solution. Table 5.1 shows the results of the investigation. Temperature of the degrees Celsius from 25 to 65 and percentage of maximum activity of immobilized trypsin, and you could see also the percentage of maximum activity of trypsin in free solution. So you could see how the maximum activity of trypsin is actually a whole lot lower compared to trypsin, even though at optimum temperatures, because obviously you could see that it's 100% active here. So at optimum temperatures we have lower activity, whereas when you increase the temperature here, the activity starts to increase all the way to 65, whereas here the enzyme has been denatured. And I think one big reason for this is because the immobilization helps to protect the enzyme from changes of pH and temperature, so it becomes just more tolerant to these changes in general. Now we can go ahead and state a reason for the difference in percentage of maximum activity of mobilized trypsin and trypsin free in solution at 25 degrees Celsius. So you can see at 25 it's supposed to be the optimum temperature for the enzyme. However, trypsin immobilized is actually being inactive, which a suggestion here should be, because it's like state a reason for the difference in percentage of maximum activity. I think we can write, for example, that the immobilized enzyme is covered. It does not react as much with the substrates. It's supposed to be that the immobilized activity is obviously going to be protected from the temperature changes. However, still, it's a disadvantage of immobilization of enzymes that the enzyme itself is in the bead, and so it's harder for the substrate to reach the active site of the enzyme. So obviously, the activity will not be as high in an immobilized enzyme compared to a free enzyme. So the enzyme active site is harder to reach in a bead compared to it's harder to reach compared to free enzyme for the substrate. So you can continue explaining by mentioning, for example, the active site, basically less enzyme peptide complexes being formed, less successful collisions, less product being released, and so on. So Justin explained why the percentage of maximum activity of immobilized trypsin at 55 degrees Celsius is higher than the percentage of maximum activity of trypsin in solution at 55 degrees Celsius. Okay, so you can see in the graph or in the diagram that at 55 degrees Celsius, trypsin in the free solution is getting denatured, whereas trypsin in the immobilized bead is going to actually have a higher activity, and it's going to reach the maximum activity at such high temperatures. So to explain this, I could say that obviously the trypsin, immobilized trypsin, is protected against the changes in temperature. Okay. And so it does not get denatured. Unlike obviously the what's it called free solution trypsin, which is obviously getting denatured at 55 degrees Celsius, and you can give a reason why. So you can say, for example, the tertiary structure is changed, and you can explain why, because the hydrogen bonds are breaking and so on. But then obviously here you can just continue by saying that immobilized trypsin, for example, is protected, and so it does not feel the temperature because it's not as exposed to the environment. Parts of it are blocked from the surroundings. And so that's the way to answer this question. Figure 6.1 shows a plant cell in a stage of mitosis. Some of the structures shown in figure 6.1 contain DNA, and use a line labeled D to indicate one of these structures. So obviously we need to label the chromosomes, which are the darkest points right here. So we can just point a line, make sure the line is straight using a ruler, and then write D. Okay, name the line, name the stage of mitosis shown. Since the chromosomes are separated and moving to opposite poles, this is definitely anaphase. And then colchicine is a chemical used by scientists to study mitosis. This chemical inhibits the organization of microtubules in prophase of mitosis. The cell shown in figure 6.1 had not been treated with colchicine. So remember, it's a chemical to study mitosis, and this chemical inhibits the organization of microtubules in prophase of mitosis. Interesting. So this obviously has not been treated with colchicine. Explain the evidence that the cell has not been treated with colchicine. So if it's been treated with colchicine, this prevents the microtubule organizing center, the centrioles, from producing and organizing the microtubules in such a way that during metaphase they hold the chromosomes at the equatorial plate, and during anaphase they separate the chromosomes and move towards opposite poles of the spindle. But here obviously we see that the chromosomes are clearly in anaphase, and they're clearly moving towards opposite poles, being held and being organized, or being held by the microtubules, which are connected to the microtubule organizing center. So it's definitely not been inhibited. So let's write this all down. In figure 6.1, the cell is in anaphase, so the chromosomes / you could say sister chromatids here are being pulled to opposite sides or poles by the microtubules, which is only possible if they aren't inhibited in prophase. And since and microtubules are clearly organized in metaphase to hold the chromosomes at the equator, so that the cell can reach the point. Or anaphase, So it's clearly hasn't been treated with colchicine. And this is the end of the paper. So this is it for this video, guys. I hope this video was useful to you. If it was, make sure to like, share, and subscribe so that this video could be shared to as many students as possible. Now I'm going to be seeing you guys in my next one, and goodbye.